Alright, welcome to video 3. I am hoping this is the last video, but I could be lying to you. There might be a fourth video. So what we're just talking about are differences in the animal kingdom that kind of helps us classify our different animals. And we'll talk about these again when we talk about the different phyla, but this is just a good introduction to explain what these different terms mean, so that when we're talking about our jellyfish or sponges or humans, that you're like, oh yeah, if I think back to our lectures, we've already talked about this. So don't worry if you're like totally overwhelmed. You're going to see this information over and over again. So we just talked about symmetry. Another way we can differentiate our different animal phyla is germ layers. Germ is referring to kind of the body, and typically when we talk about them in this way, we're talking about layers of cells during development. So you're thinking, sperm just meant egg, you have a zygote. That zygote is now multiplying to create that multicellular organism. Now, those first few cells start differentiating into different layers, or the germ layers. And this picture that you see, you don't need to know these key terms, but this is showing that zygote now undergoing mitosis. So more mitosis is getting larger. And then this one is color-coded, showing you different types of cells that have developed even very early on in the mitosis process. So we can separate our organisms, our animals, into two categories. We have diploblast and triploblast. Our diplo, di means two. So these organisms have two layers, two germ layers. So the two layers they have, first one is the endoderm. Endo referring to inside, and derm usually refers to a layer. So the endoderm is the internal layer. This typically gets churned into the lining of the digestive tract and the respiratory tract. So lining, they have epithelial cells. So this becomes the inside of the digestive system, or it becomes, the inside of the respiratory system. If you look, this is an example of a jellyfish, which are examples of diploblasts. The kind of yellow squares, that's representing our endoderm. This is lining the internal cavity. In a jellyfish, the internal cavity is essentially the gastro cavity. This is where they're digesting their food. A diploblast also has an ectoderm, ecto meaning outer. So this is an outer layer. These are outer epithelial cells. In some organisms, or in most organisms, this is also what's developing the nervous system. So thinking about your nerves and brain. Here in this jellyfish, which is an example of a diploblast, the ectoderm is all this blue-purple area, so the outer part of this organism. Diploblasts also happen to be our radial symmetric organisms. So our sea anemones and our jellyfish and related species. But that's actually a very small, percentage of all the animals. Most animals are triploblasts. So they have an endoderm. Endoderm, just like our diploblasts, serve the same exact function. Inside layer, lines digestive, lines respiratory. So that's the same. Now, the next layer is a new layer, what we call the mesoderm. Meso means middle, and this is what's lacking in our diploblasts. The mesoderm has connective tissues. It has things like muscles, or it develops things like muscles. It develops blood. It develops, cartilage and bones. So things that are missing in jellyfish and sea anemones. So we have all these different types of tissues that need to be created, and that stems from the mesoderm, or the middle germ layer. And then finally we have an ectoderm. Similar to our jellyfish, the ectoderm is the outer layer. This is our outer epithelial cells. It also develops our nervous system. So everything's the same, it's just we also have an additional layer because we're more complex. We have all of these organs and organ systems and connective tissue between all of them. So this is one or another way to classify our animals. And just like our diploblasts were all radial organisms, our triploblasts are all bilateral organisms. Now we can break our triploblasts apart in the sense that if we can further classify those organisms. So again, these are organisms that have 3 germ layers, our endo, meso, and ectoderm. So inner, middle, and outer germ layers. And the way we organize them is by their presence or absence or weird situation of a coelom. So this word right here, you pronounce that coelom. The coelom is an internal body cavity. And by this, if we think about humans, we have a thoracic cavity. This is where our lungs are. You can also find our heart in it. If you were to open up a human and not cut very deep, that whole area is actually open. Like we have space between our lungs and between our heart. There's fluid and stuff in there, but there's space. It's not just like dense tissue throughout. You have your organs and then an empty space, and then those organs are usually encapsulated in some sort of membrane. So humans, for example, we have a coelom. We have different body cavities where we find our organs. But there is space around those organs. So that's the cavity, is the space between our organs. This is a, I guess, evolutionarily complex trait. So this developed over time. And it's beneficial because it helps organs grow optimally, meaning there is open space for the heart to grow, for the lungs to grow. They're not bound by as many constraints as lacking a coelom. It also provides cushion. I mean, that air that's around your organs helps that when you get hit with something, you kind of have this cushion to kind of prevent injury to your organs, or at least prevent less injury to your organs. And finally, it also allows you more flexibility. Because we have this extra space in our body cavity, we can do things like twist. Our organs kind of reposition as they need to because they have this space to reposition, which isn't true of organisms lacking a coelom. So we have 3 different types of coeloms that we find in organisms. The first one are eucoelomates. That prefix EU means true. So a eucoelomate is an organism that has a coelom. For example, humans. Most of the animals that you learned about starting in kindergarten, those are all eucoelomates. Like most of the organisms that you're probably going to name in your head right now are all eucoelomates. If we look at this picture, this diagram, you will see a diagram like this again. First, I'll point out the ectoderm. The ectoderm is our epidermis. So my arm, this is all the ectoderm that you're looking at. Then you have the endoderm. So if I think maybe more of my stomach region, my stomach is covered in, my stomach is part of the endoderm. The endoderm is the lining of your intestines and gastro system and respiratory system. And then our mesoderm, that's going to be all of the other stuff. This was the connective tissue. This was the muscles and the blood and the bones. In a coelomate, you see that the mesoderm lines the ectoderm. So underneath your skin, you have a layer of fat and a layer of tissue. Then you have this open space. That would be your body cavity. And then you have a lining still of mesoderm. You have a lining around your different organs. For example, the small intestine has a lining of muscles. Your kidneys have a lining of fat. All of these have a lining of epithelial cells. So it's the mesoderm that is lining both the endo and ectoderm and creating this body cavity. And this is very important because you're going to see our 2 other organisms are just different. Now the organisms that this falls into, this includes annelids. Annelids are like your earthworms. This includes mollusks. So you're thinking snails, for example, echinoderms, which are our starfish, and chordates, that includes us as humans. So you can see a wide variety of organisms are considered eucoelomates. Our next organization is acoelomate. These are organisms that lack a coelom. They do not have a body cavity. Now remember, we're talking about trypoblast organisms. These organisms have an endo, ecto, and mesoderm. However, the mesoderm doesn't separate to create that body cavity. In this case, looking at this picture, ectoderm outside blue, endoderm yellow inside, mesoderm in the middle. This is where tissues are. These are where primitive organs are, but there's no space. There's no cushion that we are talking about. It's just all the organs kind of plastered right next to each other with no, air opening or anything between them. It's just an all continuous organism with no space between organs. This is pretty limited in the animal kingdom. You're only finding this in our flatworms. That includes planaria, which we're going to take a look at in lab when we get back, as well as tapeworms, so the parasitic worms that you can find in humans, but all sorts of organisms. Those are really the only two classifications that lack a coelom. And then we have these weird guys, these pseudocoelomates. Pseudo meaning false. They have a coelom, they have a body cavity. However, it's not completely lined with mesoderm. Remember with eucoelomates, that mesoderm, the connective tissue, you could find it in the ectoderm, you could find it around the endoderm. Not true with our pseudocoelomates. They have an opening. However, their endoderm is only lining this outer ectoderm. It's only lining the skin. That's the equivalent of having the fat tissue under our skin. However, around their digestive tract or respiratory tract, there's no protection. There is no mesoderm to help protect it. So yes, there's an open body cavity, but there's zero tissue around the gastrosystem, around the respiratory system. And you can see that can kind of cause problems with just, one, collapse of organs, but two, just less protection because you don't have a layer of protection around your organs. This very limited in the evolutionary tree. We're only finding this in our roundworms. Roundworms, they're mostly parasitic. They cause different parasitic diseases both in humans as well as, for example, heartworm. Heartworm found in dogs are caused by roundworms. So they're a very small portion of the evolutionary tree. Alright, so I lied to you guys. There is going to be a fourth video, but only a couple of slides on it, and I'll also explain what's coming up in the next video as well. So I apologize. There is a fourth video, but it will be a very short video. Alright, see you then.